Okay, so ladies and gentlemen, now the screen because of the cable.
Wow.
Yeah, it's kind of. Oh no.
There.
Okay, so thankfully. Now, so we're gonna. So yesterday, as you saw, we kind of halfway done the meiosis. Okay, so yesterday, and I'm just gonna quickly go over. So yesterday we kind of gone over the first half of meiosis. Okay, so you have the homologous pairs lined up. Remember, in human, you have 23 pairs of chromosome. So, for example, in this diagram that you guys are going to see later on, yeah, so we would have in our regular body cells would have 23 pairs. So 22 pairs would be what you call autosomal. Regardless whether you're a boy or girl, you have those 22 pairs. And the last pair is the sex chromosome. So if you're a girl, you have the 2XX chromosome. If you're a boy, you have the XY chromosome. Now, homologous pairs, just like you see, you have a pair of chromosome number 1. If you compare all the chromosome number 1 to other chromosomes, they are different length, and the band pattern that you notice, they're different between different chromosomes, but they're the same in between the two homologous pairs. Do you guys see that? Okay, so that's what we mean by homologous pairs. So they line up, they pair up together, not line up, they pair up. Now this is the crossover situation happens. Portion of the chromosome that would be from dad will go on to the part that is from mom and vice versa. Okay, these crossing over is one source of genetic diversity. Okay, so you shuffle your deck once. Meiosis, what is it? Metaphase 1, you have these chromosomes line up along the middle. Okay, the orientation is completely random. So we call that independent assortment. So that's another source of genetic diversity. Anaphase 1, these chromosomes separate to both their sides because their microtubule contracts. Telophase 1. Telophase 1. This is the part where you start going to have two cells. Each will have 23 chromosomes, but you have 46 chromatids. Okay, you still have double the genetic information that you want in a gamete. So that's where we were at yesterday. Any questions so far? Okay, so we're going to continue on in terms of the diagram. Okay, so prophase 1, prophase 2 now. We're going to be at prophase 2. So again, these chromosomes have condensed in the middle. The nuclear membrane has dissolved in both cells.
Are these still touching? Or?
No, they're just close.
Oh.
Remember, I fold the paper in half, so I only have that much space. Yeah. So prophase 2, these chromosomes are in the middle on their own. The nuclear envelope dissolving. And metaphase 2, Yes.
What's into each type? Is that not just cytokinesis?
Yeah, previously there's a step called cytokinesis in between here. I just didn't draw.
Oh, so then what's happening in prophase 2?
So what happened in prophase 2, after the cytokinesis, there would be another interface. So there would be an interface between these two, and the chromosomes will be separated. There would be a nuclear envelope in between each, and then another checkpoint happened. Then the nuclear envelope dissolved again. So it's really mitosis happens twice. You have two interphase, two prophase, two metaphase, anaphase, telophase, and two cytokinesis. Okay. Now put up your hand if you're done. If you're done, okay? Both of them. Okay? When you're done both of them, let me know.
What does it say over there? Due to
Random.
Due to random orientation. So here, another independent assortment due to random orientation. Yes. Okay. You're done?
Mr. Chang,
Yes.
what do the dashes in
This here?
like yeah? What do
That's the dissolving nuclear envelope. Okay, put up your hand if you're done. Okay. So I'm just gonna talk about this part here for those to give you a chance for those of you still catching up. So here in metaphase 2, these chromosomes are lining up again along the equatorial plate or along the equator. Now again, the orientation of these chromosomes are completely random orientation. So in this case, it will result in what you call another independent assortment. Okay, so this will be the third time during the whole process of meiosis that the deck got shuffled, so to speak. Are we good on that? Okay. Now, anaphase 2. So anaphase 2, the microtubule contracts and the sister chromatids separate from the chromosomes. Now, these sister chromatids are now you call them chromosomes now. So it's no longer sister chromatids, but you call them chromosomes. Because under the microscope, they are distinct blobs. Yes.
Can you repeat what you said for metaphase 2?
So metaphase 2, they're all lined up along the middle of the cell, the equator of the cell. The arrangement, the orientation is completely random. So in this case, it's almost like shuffling the deck a third time. Okay, so another independent assortment due to random orientation. Okay. Any questions? So I'm just going to pull this a little bit more to the side. A lot of people still drawing, so I'm going to give it a few more minutes. Okay, put up your hand if you're done. Both. Okay. Give 2 more minutes, I guess. Who's done? Okay, most of you. Now, as you see here, by the end of meiosis, so this one I have telophase 2 as well as the cytokinesis. So the end product, you have four haploid, single small n, gametes with 23 chromosomes each. Now, here, remember, I only show 2 out of 23 pairs of chromosomes. But again, due to crossover and 2 events of independent assortments, okay, 2 events of independent assortments, you can see all of these 4 gametes are different from each other, okay? And there could be other more combinations too, simply because the orientation could be more flipped different ways. So in this case, this meiosis provides it's like one huge mechanism for providing like that genetic diversity, which sexual reproduction allows, okay, because of this process. Any questions so far? No? Okay. Now, typically, in the next day or two, I would be quizzing my students, like a short 5-minute quiz on process of meiosis. Okay? So make sure you look over these things and study them. Okay? We're gonna go through the slides because remember, on the drawing, I didn't write the blurbs down that much. So we're gonna look at the slides. Okay? So we ended around here, right? So we ended around here. No, we started around here. Meiosis interface, prophase 1, prophase 2, metaphase 2, 1, anaphase 1, telophase 1, cytokinesis. This is where we stopped yesterday. Yeah, so in prophase 1, as you saw earlier, the nuclear membrane dissolved. Now, I didn't draw it on the diagram, but here, as you can see, the spindle fiber starts to form again. Okay, they were dissolving at metaphase 1. So start to form, all lining up in the middle, and sister chromatids are still attached to their centromeres in metaphase 2. Anaphase 2, as I said, the centromere split. Now the sister chromatids are separated to move into the opposite poles because these are now distinctively their own blob under the microscope. So these sister chromatids are now called chromosomes. Last but not least, you have now 4 gametes. Okay, each half of the chromosome numbers of the somatic cells. Okay, regular body cells. Any questions so far? So make sure you know the blobs. Make sure you know the different writing on each phases too. Okay. So these are the diagrams basically comparing both mitosis as well as meiosis. Okay. Any questions? Okay. So I'm gonna stop here. We're gonna do our activity now.